All right, so today we are jumping in to the topic of blood typing. Now, before we get into um, blood typing itself, we need to discuss the different types of blood types, right? So, with that, there's two types, or two type groups. So, say, um, so you have your AB group and then the O group. Now, if you're in the AB group, we can have A positive, A negative, B positive, and B negative. Also, we can have AB positive and AB negative. Okay? So in the O group, there's only two of these bad boys. You either have O positive or O negative. Okay? Simple, right? So that is all of the blood types. That is every single blood type um, that you can possibly be. So now that we know that, we know we have two blood typing groups. You're either in the AB group, AB category, or the O category. And if you're in the AB group, you're either one of these. And if you're the O category, you're one of those, right? Simple enough. So, how do we know what blood type we are? Well, there's two factors in determining this. There's the antigens and antibodies. Fix my little E there. Well, first we're going to talk about antigens. So, antigens are the name tags that tell us what our blood type is. So, I guess we can look at it like this. So, on our red blood cell, so let's get us a red blood cell here. It's a weird blood cell, but okay, it'll do. So, this is our red blood cell. We're going to have these things called antigens that tell us our name, right? Antigens are found on the surface of our red blood cells. So if you have, and the only antigens that are possible, so let's write this back up here, antigens, you can either have A antigen, B antigen, and RH antigen. Now, RH is special because RH is what codes for if our blood is positive or negative. So if we have the A antigen, we know that we have a type A blood. If we have B antigen, we know we're type B blood. Now, if RH, and we'll do RH in green. So if RH antigen is present as a name tag on our red blood cell, then we know that we are positive. Now, if we don't have the RH antigen on our weird red blood cell here, that's going to signal that we're RH negative, which means that we have a negative blood. So for an example, let's say that we'll use A positive blood, okay? Just for a quick little example here. So here's our red blood cell, and we're looking for our antigens that we have. So we'll use orange for B. We will use, I don't know, let's use purple. Purple for A. And we're trying to find it for A positive, right? So for A positive blood, we're going to have the antigens that signal for A, and we're positive, so we need that RH antigen as well. So by looking at this red blood cell here in our example, we see that we have the A antigen and the RH antigen, which makes our blood A positive. Okay? Okay, I hope everybody is following along so far. So let's do another quick example here. Let's say that, okay, so our red blood cell, and let's do, here's another antigen. So what would this blood type be? If you had a blood cell, a red blood cell, and you've seen these 3 antigens on the surface, because that's where antigens are found. They're found on the surface of our red blood cells. So if you've seen these on the surface, what is that going to signal for us? Well, that's going to mean that hold on one second. Sorry, I had a technical difficulty. So if we have these antigens, that's going to mean that our blood is A, because we have our A, B, because we have the B one as well, and positive, because we have our H. So now that I feel comfortable about that, we'll move along. Okay. So if you can remember, the next factor that we have to account for is the antibodies. So what are antibodies? Well, antibodies, I like to think of them as the fighters. So they fight. So if you have A blood, you can't get B blood. If you were given B blood, what's going to happen is called an agglutination, which means your blood's going to clot, and that's going to be very deadly very quickly. So when antibodies are going to develop in response to the antigens that you have. So let's use an example so we can see this a little clearer. So let's say that we have A positive blood. Now, side note here, positive blood can get positive or negative. Negative blood can only receive negative blood. You can't give a positive to negative, but you can give negative to positive. Okay? So keeping that in mind, if we have A positive blood, which means that we have the A antigen and the RH antigen, we're going to develop antibodies that will fight off B blood and, well, just fight off B blood. So the antibodies that we would have are anti-B antibodies, which means that anytime you are given B blood, these fighters, anti-B is anti-B, and it's going to come in and fight it. So if you have B negative blood, the antibodies that you would have would be anti-A and anti-RH, which means it's going to fight any A blood and it's going to fight any positive blood. So let's go in and let's say, let's just make us a little chart here. So we have, I need to turn my eraser off. We have A positive, B positive, A negative, AB positive, AB negative, AB positive, AB, well, let me fix that. Positive, AB negative, O positive, and O negative. So let's just chart out what blood can be received from each, or what antigens and antibodies that we'll have. Okay. So for our antibodies, we're going to write them in antigens. We're going to do in purple, and antibodies we'll do in red or green. Okay. So for A positive blood, we're going to have A antigen and RH antigen. We're also going to have anti-B. For B positive, we're going to have B antigen and RH antigen, and anti-A antibody. Okay, A negative, so we have the A, and we do not have anything else. We're going to have the anti-B and anti-RH. AB positive will have A, B, and RH. Now, what antibodies would A positive have? None, because it can take everything. This is called the universal recipient. Because it can take blood from anyone. So that's a very cool blood to have, because it doesn't matter whose blood they give you, you'll be okay, because you don't have any antibodies that will create that allergic reaction to cause the clotting. All right, let's jump back in. So AB negative. AB negative is going to have A, B, and nothing else. And it's going to have anti-RH. O positive. So with O's, there's no O antigen, right? So that just means that it doesn't have any antigens, but since it's positive, it does have RH. So if you see a red blood cell that only has that RH antigen, you know it's O positive. O positive is going to have anti-B, RA, and anti-B. So we also have O negative here. So if it doesn't have A, it doesn't have B, and it's not positive, what does that mean? It has no antigens, none. So this is our universal donor. Since it's our universal donor, that means it can give blood to anybody, because it doesn't have any antigens on it that will counteract with these antibodies here, creating an allergic reaction. So O negative blood is like, if you have O negative blood, the blood banks are probably calling you all the time. You can donate to everybody, and it's awesome to have, because then the hospitals don't even have to do blood testing or anything on you. They can just give you O negative blood and be fine, because anybody can receive it, which is pretty cool. But one thing that's not cool about O negative blood, since it doesn't have any antibodies, it can only receive itself. because since it doesn't have any antigens, I'm sorry, I said that wrong. Since it doesn't have any antigens, it can only receive itself. So it's going to have anti-A, anti-B. I'm just going to write it under here. So anti-A, anti-B, and anti-RH. So the only thing that it can receive is O negative. So let's do a little practice run through real quick. Okay. So if you have someone with AB negative blood, what blood can they receive? If you want to take a second and pause the video and try to work it out and try to remember it, then pick it back up. So the blood that it can receive is A negative, B negative, and AB negative. Oh, and O negative, because that's our universal donor. So that is the only blood types that this one can receive. Okay, let's try another one. All right, let's try A negative. A negative can receive O negative and A negative. That's it. All right, let's move along. So now we're in the heart of blood typing. And what I say about that is the ever dreaded agglutination test. So let's think for a second. Let's just put ourselves in this scenario. You've been in an accident, or either you're needing blood for some reason. So you're in the hospital, and the emergency room staff, medical professionals here, right? They don't know what type of blood you are. And they can't just guess at it, because again, that could kill you. So they need to test your blood to see what type you are. So what they're going to do is they're going to take a sample of your blood, bring it on back to the lab, and back in the lab, they're going to have a tray with 3 little test tubes. Now, in these test tubes, there's going to be solutions, a solute that has antibodies, okay? So this will be our A antibodies, and we'll have our B antibodies here. And then we'll have our RH antibodies. So when they get back there, they're going to take your blood and they're going to drop it in to all 3 of these to see. And what this is testing for is this is testing to see if a reaction occurs. Why did I just do that? Let's put that back on there. Come on. My eraser's too big. Okay. So let's say, and with the reaction, what I mean by that is, since these are antibodies, and let's just write that. Well, we need a marker to write that, so let's just write that. These are the antibodies. So since these are antibodies, they're what? They're fighters. So when you drop the blood into the antibodies, you're going to either, so if it's anti-A antibodies, it's going to fight what? It's going to fight A blood. So if you're dropping A blood into it, it's going to agglutinate. If you're type B blood, and I drop your blood into this test tube here, the anti-B antibodies are going to fight B blood. So if you have B blood and I drop it in here, it's going to clump, it's going to agglutinate. The reaction is going to occur. Same thing with this one. Same thing with RH. So let's do a little example. So we bring blood back to this little lab, in our imaginary lab here, and we have our test tubes. Let's draw some new test tubes. Let's just make little circles, okay? So we have our test tubes, and this is going to be our RH, and it's always going to be labeled like this. You're going to know what antibodies are in there, because without that, how are you going to know what's going on? So they're going to be labeled, and we'll write it off to the side here so that you can keep it in mind. These are the antigens. Okay, so I have a patient's blood, and I'm about to drop it in. So when I dropped it in, I had an agglutination here, and I had an agglutination in this one. So from that agglutination, what is this person or this patient's blood type? Well, since they had an agglutination in the B, we know that they're B blood. And since they had an agglutination in RH, we know that they're B positive. So now we can go back to that patient, and we could either give them B positive blood, or O positive blood, or O negative blood, right? Or we can even give them B negative blood. So we know now, we've narrowed it down, and we know what's safe to give them and what's not safe to give them because of that reaction. There was no agglutination in the A for B positive blood because this is anti-A, and it fights A blood. So since we don't have A blood, there wasn't an agglutination. The reaction didn't occur. Okay, let's try another one. Let me get this erased here. All right, let's put our test tubes back up there and our labels. Okay, all right, so I have patient number 2's blood, about to drop it in. All right, agglutination. Okay. All right. So look at all of these agglutinations. What does this mean? So let's look at it, and let's determine, now that we've done the test, we can tell what our patient has. From this test, we know that there was agglutination in everything. So we have AB positive blood. So patient number 2 has AB positive blood. So what does that mean? Well, that tells us that we can give that patient anything. Why? Because AB positive is the universal recipient. So we're safe to give that patient anything that we have on hand and that's available. Okay. So now that we've covered all of our bases here, I hope that you feel more comfortable with blood typing, and I hope that my explanation was helpful to you. And I'm just going to write these terms again for you. So let's just start off here with the marker because I don't need the eraser. So antibodies, or antigens. So antigens are the name tags. They are going to be found on the surface of red blood cells. And from the antigens that we have that are present, our body, is going to produce our antibodies, which are our fighters. So they're going to aid in immune responses. These are called immunoglobulins. Immunoglobulins. So we have our antigens and our antibodies that tell us our blood type. And now we know how to do the agglutination test. And agglutination is just a fancy word for what? Blood clotting. Which is an allergic reaction, an immune response that happens when we receive the wrong blood type, right? Which is deadly. Okay, so now that you're masters in blood typing, we're going to end the video. See you next time. Thanks.